Hello. So I'm Mrs. Jones, and I'm going to give a couple minutes for everyone to get in or whoever's planning on joining us today. So this is a Unit 1 review, and so it's meant for us to be able to go over the various topics from Unit 1. My students have a test coming up this week, so I figured this would be a great opportunity for us to go ahead and do a Unit 1 review for any of those AP Bio students out there or regular biology students that are out there. So I believe there's a way that at the bottom you guys can write questions if you have any questions. So first thing I'm going to do is just say what overall Unit 1 is about. That way it gives you guys a chance to be able to put your questions at the bottom. There should be a little question mark. And if you type that, you can type a question in there. And I'm going to plan on doing this for about an hour or whenever the questions stop. So if you don't have any questions, then of course I will just end the Instagram live because I will have made the goal, which was to answer your questions. This is actually my first solo mission. If you saw the absolute recap down there, she just wrote hello. At the end of last school year, when we were reviewing for the AP exam, her and I did a bunch of co-workings on this so that we guys work together. So if you're not following the absolute recap, be sure that you do follow her. She does a podcast for AP Biology. She also does podcasts for chemistry and physics and music history, and I believe there's world history, all kinds of stuff. Hi Marker Learning. So real quick, Unit 1 is about just overall bonding rules because that's the foundation for a lot of the stuff that we're going on this year. Also, it has to do with water properties. So all the different properties of water based on hydrogen bonds. And then, of course, the macromolecules. So looking at carbohydrates and proteins and nucleic acids and lipids. So that is what Unit 1 is about. So does anybody have any questions? As of right now, there's been no questions that pop up, y'all. So I'm running this based on you guys. So I need you to ask questions on what you want me to review or what you're needing information on. So again, type it into the little question thing so that I can help you because I need to know what you want from me. If you're not going to ask the questions, then I'm going to end the Instagram live. Questions? Anybody? Okay. So I guess I'll just ramble for a little while since no one's asking questions. So there are four main macromolecules. The four main macromolecules that we have lipids. Lipids are the main one that's nonpolar. And there's three different types of lipids. We have steroids, and the way that I always remember steroids that there's four ring structures, so it's four rings put together, and of course nonpolar. And then the the cholesterol tends to be the starting material for all of the steroids. And because of the fact that they're nonpolar, when we get into cell communication, we're going to talk about how the receptor for a steroid is going to be inside of the cell. We also have phospholipids. Phospholipids is another type of lipids, and they're made by having a phosphate group and then two fatty acid chains attached to them. And so the advantage of having a phospholipid is that that phosphate group, because it's charged, ends up being polar. And so because of the fact that it's polar, it will face the water. And then we're going to have those hydrophobic tails because they're fatty acids. And they're going to face inwards. And so it kind of looks like we've got our phosphate heads on the outside and our fatty acid tails on the inside. And it's going to create a membrane. And if you just joined us, I'm just kind of going over lipids real fast while I wait for some people to answer write some questions down there. So if you're having questions, be sure to put them down there. And then the third type of lipid that we have are fats. And fats are called triglycerides because of the fact that they have a glycerol molecule, and then there's three fatty acids that come off of them. And because of those fatty acids, these are non-polar substances. So I see that I've got two questions, so let's see what those questions are. Okay, so let's see. How can you tell the difference between amino acids? And so I believe that this student is probably asking about polar versus non-polar versus charged side groups, because amino acids have the same general structure. They're always going to have the amino group, and the amino group is going to be basic. It's NH2, unless we're in the ionized form, then it's NH3. And then the other side of it, we're going to have a carboxyl group, which is COOH, or again, if it's ionized, it's COO minus. And then there'll be a hydrogen. And then there's this R group, and that R group can change. And so if we're looking at a polar side chain or a polar R group, that means that we're going to have a lot of oxygens or sulfurs in there, because the oxygen to carbon bond or the oxygen to hydrogen bond is a polar covalent bond. And because it's polar covalent, it's going to make that R group be polar. The nonpolar side chains, or the nonpolar R groups, are going to have hydrocarbon tails. You're going to see the ring structures in there. You'll just see a whole bunch of carbons and hydrogens. You'll see branches of carbons and hydrogens. It's going to be made of almost all carbon and hydrogen if it's a nonpolar side chain. And then if it's a charged side chain, that means that we're going to have a positive or a negative charge on it. It'll be kind of an ionic bond. And so what that does is that causes also a polarity. And so our polar and our charged side groups are going to aim outwards, or they'll fold outwards so that they face the water, versus the nonpolar side chains are going to fold inwards because they're afraid of the water. They're hydrophobic, or they're afraid of the water. So they kind of fold inwards to kind of hide away from the water, while the hydrophilic, which would be your polar and your charged side groups, kind of face outwards. So I hope that answered that person's question. It looks like there's one more question down here. Marco Learning is asking me some questions about the AP exam. Do I think the AP exam is going to still be the full 3 hours? Yes, I do think that. From everyone that I've spoken to at College Board, it looks like that they are planning on the normal exam. I have heard a little bit of inklings of that, depending on your learning environment this year, that they might be doing a modified exam at home. But based on the fact that the PSAT is actually being taken place in all these schools, like my school is still going to do the PSAT even though we're 100% virtual this year. So I do not see any reason why my students won't have a full exam, and I don't see any reason why some of these other schools won't have full exams. So I do believe it'll still be the full 3 hours. You'll have the 90 minutes for your 60 multiple choice questions, and then you'll have your 90 minutes for your 2 long and 4 short for response questions. But this is supposed to be about Unit 1. Let's see what other questions we have down here. What kind of resources do you have on your page? On my page, this is just a review account, and so what I've been doing is that, starting in February, I'll start to do a big review for the AP exam. Every single day, I'll post in my story questions. If you've been following me the past week, last weekend you saw the review questions I posted for my students, and then this weekend you also saw the review questions I posted for my students. So I post those kind of before the AP exam. You actually can see all the ones from last year up there again. They'll probably be very similar questions again this year. And then I'll do a bunch of Instagram lives. I'll probably convince my friend, the Absolute Recap, to do it with me again. So let's see, do we have any more actual content questions? Okay, no. So other than lipids, I guess we could talk about another one. Yes, we do have the best FRQ reviews. So another one of our macromolecules that we have are nucleic acids. Nucleic acids monomer is going to be a nucleotide. And so the nucleotide is made up of 3 components. You have a phosphate group, you have a pentose sugar, and then you have a nitrogenous base. And so there's 2 main types of nucleic acids that we have. We've got DNA and we have RNA. DNA's pentose sugar is going to be deoxyribose, while RNA's pentose sugar is going to be ribose. And so there's one difference between the 2. When we talk about how many strands they have, DNA is traditionally going to be a double-stranded molecule, while RNA is going to be a single-stranded molecule. And then when we talk about the nitrogenous bases, DNA is going to have adenine, thymine, cytosine, and guanine. And adenine and thymine will bond together, and cytosine and guanine will bond together. Okay, so I always tell my students at Conyers, Georgia, because of the fact that our school's in Conyers, Georgia. So I say at Conyers, Georgia to help them remember ATCG. And that's with DNA, of course. And I went to Georgia College, and so when I was in college, the way I remembered it was at Georgia College. So you just kind of find a way to help yourself to remember A and T, C and G. And then when we're talking about RNA, RNA is only going to have the adenine, a uracil, instead of a thymine, cytosine and guanine. And so whenever you're looking at multiple choice questions in which they're asking you to give an RNA molecule, if you see any thymines in there, you immediately know that that answer choice is not a good answer choice, and you can immediately kick that one out because of the fact that we will only find uracil in RNA, and we're only going to find thymine in DNA. The directionality is important to know about nucleic acids. Nucleic acids are, there's a 5' end and a 3' end. The 5' end is going to have a phosphate group on it, while the 3' end has a hydroxyl group on it. And so what that means is that if you were to number the carbons in a sugar, because we have a pentose sugar, the 5th carbon is going to have the phosphate attached to it, while the 3rd carbon has a hydroxyl group attached to it, and that's why they have the 5', 3' ends to it. The DNA, as we said before, was double-stranded, and so it's going to have this anti-parallel structure. And so you'll have a 5' end that will pair up with a 3' end, and then a 5' end that will pair up with a 3' end. And so that's a quick review on nucleic acids. Remember, if you have questions, put them in the little box down there. I would love to answer your questions instead of rambling about the macromolecules. And the macromolecule that we have would be the carbohydrates. Carbohydrates are going to be your sugars. And so these are made up of carbon, hydrogen, and oxygen. And if we're looking at a simple sugar, it's usually a 1 to 2 to 1 ratio. And so an example of a simple sugar would be glucose. And you're going to do a lot more with glucose when you get into cell respiration in Unit 3. But so the big thing about carbohydrates is being sure that you understand the differences between them. So if we're talking about cellulose and starch, cellulose and starch are both made in plants. The starch molecules you're able to break down because they're alpha linkages, versus the beta linkages in cellulose that is in the cell wall of a plant, you're unable to break down. So a lot of times we have these organisms that consume plants. So for example, a termite. A termite eats wood. And so you're thinking to yourself, well, if a termite is unable to break down wood, why would it consume wood? Well, the termite has a microorganism that lives in its intestines, and that microorganism is going to break down the cellulose. And so by breaking down the cellulose, it's going to go through a hydrolysis reaction to release out these different small molecules of glucose. And so this glucose can then be absorbed by the intestines, and so the termite is going to get food of glucose, while the microorganism also gets food by breaking down that cellulose to also get that glucose. And then other things to make sure that you understand about our carbohydrates is that just because there is a carbohydrate in a other macromolecule, it's not always a carbohydrate. So nucleic acids, we said before, had a pentose sugar. Yeah, there's a sugar involved in it, but as soon as you see that sugar, don't just hone your brain in and go, oh, it's a carbohydrate, because there are also carbohydrates in other components. Also, when you're creating bonds, be sure to remember that you're taking out those water molecules. So if I put together 2 glucose molecules, glucose is C6, H12O6. So logically in your brain go, oh, well, if I put 2 of them together, I have C12, H24O12, because of the fact that I just multiplied by 2. But in a dehydration reaction, you take a water molecule out. And so it would actually be C12, H22O11, because you have to take out one H2O. And I see I have a question, so let's see what they need. How much emphasis is on Unit 1 in the exam? I can't remember off the top of my head, but Unit 1 is about 18%. If I was a betting woman, I believe it's about 18% on the AP exam. And so the questions that you're going to find that are Unit 1 are going to be more of the lower level questions. They're just the straight, what are the components of a macromolecule? What are the differences between DNA and RNA? They're very low-level questions because they haven't really amped it up yet. The higher level questions we're going to find will be in our later units. So they'll be asking you specific questions about DNA and RNA, but that will be embedded within a Unit 4 type question because it will be talking about replication. And so it won't be specifically macromolecules, but these macromolecules are actually the entire year long. We're going to use these macromolecules when we talk about replication and transcription and translation. We're going to talk about them when we talk about cellular respiration and photosynthesis. We're going to talk about them when we talk about genetics. We're going to talk about them when we talk about basically all year long. Like we talk about evolution. and about how an organism is able to show one trait. Well, that's because of the fact that in their DNA, they have that trait, which makes a certain protein, which then gives you a physical trait that's seen by nature. So although it's only 18% of the exam, it's actually going to be the foundation for a lot of the other components. So I hope I answered your question. So we're again back out of questions. So remember, if you have a question, please put it in the little question box at the bottom. And I'm only going to basically ramble until I run out of ramblings, because I'm waiting for y'all to ask some questions. So we've already talked about carbohydrates, we talked about nucleic acids, and we talked about the lipids. So the last macromolecule we can talk about is proteins. Okay, so proteins, as we already mentioned with the earlier question, these are made up of amino acids. And amino acids are going to have, of course, the amino group and the carboxyl group and the hydrogen. And then they have that R group that's going to change. There's 20 different R groups. Now, you're not going to be required to know in the AP exam the different R groups. What they'll do is they'll either give you a structure, or they'll describe it to you and they'll tell you what that R group is. And then they'll ask you questions about it. And so, for example, earlier someone asked about the difference between polar and nonpolar and the charged. And so they may tell you that there's this side chain that's CH2, CH2, CH3. And they're going to say, well, how is this going to fold? And you're going to have to look at that and say, okay, well, CH2, that's a nonpolar bond. And you're going to have to think, that's going to fold inward because it's going to be hydrophobic. So that's the type of questions that they can ask you in terms of the R groups. They can also ask you about the bonding of it. So when you go through a dehydration reaction and you put two peptides together and you make a dipeptide molecule, you're always going to make that bond between the amino of one group and the carboxyl of the next group. Okay, and so what that ends up doing is it creates an N-terminus, or the part of the protein that has the amino group on it, and a C-terminus, which is the part of the group that has the carboxyl group. And I've actually seen this question before where they gave you a diagram and they asked you to pick out which of these is the correct orientation for a dehydration reaction. And you had to pick out the one that showed the amino group and the carboxyl group bonding to make the peptide bond. And the peptide bond is that bond that we find between amino acids. Now, proteins have 4 levels to folding, okay? Now, the first 3 levels are involving one polypeptide, while the 4th level involves more than one polypeptide. And I see I have a question on there. I'm going to finish my rambling about the proteins, and then I'm going to go to that person's question. So, the primary structure for it is going to just be your string of amino acids. So you just have this long chain of amino acids, and they're bonded to each other by their peptide bond. So the bond that you find in the primary is a peptide bond, which is a type of covalent bond between the amino of one group and the carboxyl of the next one, of the next amino acid. Our secondary structure is going to be where you start to find alpha helices or beta pleated sheets. Alpha helices look like a coil, while beta pleated sheets look like a paper that's been folded into accordion style. And so it kind of makes this little ripple. And it's due to hydrogen bonds in the backbone. And so what's happening is that there is a hydrogen bond between the amino of one amino acid and a carboxyl of another amino acid that's really, really far away. Okay, so it's not directly next to each other. They're kind of downstream from each other. And so because that hydrogen bond, it causes the protein to loop back, which makes your alpha helix, or it can make that beta pleated sheet. So as I said before, primary is going to be the peptide bonds, string of amino acids. Secondary is going to be the hydrogen bonds in the backbone, making alpha helices or beta pleats. Tertiary structure is going to be your final 3-dimensional structure for one polypeptide. It's just when all of the kind of R groups are now interacting. And you can have any type of bond. You can have a covalent bond, an ionic bond, a hydrogen bond. You can have van der Waals forces. You could have hydrophobic interactions. You can have basically any type of bond. And so this bonding is occurring between those R groups. So as I said before, if it's polar or non-polar or charged, that's going to create this final folding for it. We'll have any of our polar groups kind of facing outward, while our non-polar groups will kind of face inward. And so that will kind of help to create that 3-dimensional structure. Now the 4th level, okay, or a coronary structure, this has to do with when I have more than one polypeptide. And so, for example, collagen. Collagen is made up of 3 polypeptides. So these are 3 polypeptides that kind of coil together, and they make this, it almost looks like licorice, this long chain of all the collagen kind of intertwined together. And so that would be an example of a coordinate structure. Another example of a coordinate structure would be hemoglobin. It has 2 alpha subunits and 2 beta subunits, and they're also going to interact together to make this big structure. And so what we kind of think about is that all proteins are polypeptides, but not all polypeptides are proteins. And so that collagen, when you just have the single polypeptide, it's not making up the full protein yet. You need those other 2 subunits in order to fully have your collagen molecule. So I think I've answered my ramblings on proteins. Let's see what this person's question is. That is a teeny tiny question. When looking at pictures of macromolecules, would you have to look at the various elements involved? And I can't actually see the rest of that question. It doesn't really, it's teeny tiny. Would you have to look at the various elements involved to identify it? Well, this is a great question to think about. So first thing we want to think about is what elements are we going to find in each of our macromolecules? So if you think about carbohydrates, carbohydrates have carbon, hydrogen, and oxygen. And those are the only things it has in it. Versus our lipids, if you talk about steroids or if you talk about your fats, those ones also are carbon, hydrogen, and oxygen. Now, the carbohydrate had a 1 to 2 to 1 ratio. And so as we said before with glucose, it's C6H12O6. So it's 1 to 2 to 1. Versus when we think about a lipid, like a fat, we're looking at a 1 to 2 to very few. And when I say very few, I just mean there's very few oxygens in it. You've got this really, really long hydrocarbon tail of carbons and hydrogens. And at the very end of it, you've got that carboxyl group, which makes the fatty acid. And so there's just a couple oxygens in there. There's really not enough to really say a lot. And it doesn't really make the nice ratio. So we say 1 to 2 to few. Now, when we talk about proteins, these are going to have carbon, hydrogen, oxygen, nitrogen, and sulfur. Okay, and so when we talk about Hershey and Chase's experiment in unit 4, we're going to think about that what they did there is they were tracking the sulfur so they could track the proteins. And so we always want to remember that sulfur is found in your proteins. And then when we think about our nucleic acids, that's going to be carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay. Again, in the Hershey and Chase experiment, they were tracking the phosphorus so they could find the nucleic acids. The whole point of Hershey and Chase was they were trying to figure out what the genetic information was for an organism, and they were able to prove that it was DNA. But we're going to talk about that later on in life. So I believe I've answered that person's question. Big thing to take away is that nitrogen is only found in proteins and nucleic acids. Sulfur is only found in proteins. Phosphorus is only found in nucleic acids. Okay, so nitrogen is in both nucleic acids and proteins. Sulfur is just in proteins. And the phosphorus is only in nucleic acids. So I hope I've answered that person's question. And I am out of questions. So, unless you have any more questions that you would like to ask me, I'm done rambling. Because I was doing this for you guys, for any questions that you guys had, to try to make sure that I was supporting and helping however best possible. I can ramble about water properties for a little while you guys try to find some questions. So water properties, water is going to be made of 2 hydrogens and an oxygen. The bond within the water molecule is considered a polar covalent bond, meaning that the electrons are unequally shared. And so because they're unequally shared, the oxygen has a partial negative charge. And so if it comes near another water molecule that has a partial positive charge on its hydrogen, there's a hydrogen bond that forms between these water molecules. So within the water is a polar covalent, and between the water is a hydrogen bond. And so what ends up happening is that they're attracted to each other. And so this is going to cause a lot of different properties with water. The first one we have is cohesion. This has to do with when a water molecule is attracted to another water molecule. And then we also have adhesion. Adhesion is when a water molecule is attracted to another polar substances. So you guys see this when we're looking at water inside of a graduated cylinder. You see that the water kind of sticks together, making the surface, and you kind of see that it climbs the sides. The climbing of the sides is showing the adhesion, while the water kind of staying together is the cohesion part of it. It also allows there to be water having a high specific heat. So it takes a lot of energy in order to change the temperature of water by 1 degree Celsius. And this is very important. And of course, maintaining body temperature is important for maintaining the environmental temperature. So think about a coastal region. The temperature stays about constant because of the fact that all day long the heat is being absorbed by the water. And then at night that heat it's kind of released back out into the atmosphere and back out into the air. So it kind of keeps it about constant versus in the desert, super hot during the day, super cold at night because of the fact that there's nothing really to absorb. Well, it doesn't really absorb the heat. The soil can't absorb it very well. We also see that water is less dense when it freezes. This means that it actually expands. And so by expanding and kind of forcing there to be these space between the water molecules, they take up more space. Thus, of course, it is less dense, and so it's going to float. Water is actually the most dense when it's at 4 degrees Celsius. And this is going to protect a lot of our wildlife. If you think about a lake, the very top of the lake is going to freeze over. And then the animals that live below it are going to be able to stay alive all winter long because, number one, there's a temperature buffer. The wind can't get to the water to cool it down anymore. And the air can't kind of cool it down. And then also, these organisms aren't going to freeze over all summer long because the water stays liquid, of course. We also have, with high specific heat, that evaporative cooling. So when you get hot, you sweat. That sweat then runs all over your skin, and then your skin is going to heat up that water, then, of course, evaporating, which takes the heat away with you. And water is a universal solvent because of the fact that it's polar, so it will dissolve other polar substances and ionic substances. So the only thing it can't dissolve is, of course, nonpolar substances. So, considering the fact that no one's asked any questions. Ooh, there's one. What did you say allows for starch to be broken down? We have the enzymes to break down starch. Starch is an alpha linkage. And so we're able to break those. We have the enzymes. You don't really need to know what enzyme it is that breaks it. What you need to make sure you understand and know is that alpha linkages can be broken down and beta linkages cannot. Starch has alpha linkages, cellulose does not. And if you really care, you don't really know this. The alpha linkages has to do with the fact that you've got your CH2OH molecule across the top of all of those structures versus in the beta linkage you see that it flips because of the way that that bond is formed. And that has to do with why we can't break it is because of the flipping of that glucose molecule every other turn. Any other questions we have in here? Okay. So, I hope you guys have found this helpful. Give you 5 more seconds to write any questions if you have any. And I hope you guys have a wonderful day. And I hope this was helpful. And of course, I hope you keep following and let me know if you have anything. Hey, there's 2 more questions. Y'all are good at this whole let's write questions. Can you say what we need to remember about the methyl group again? Sure, we can talk about functional groups. So the methyl group is a CH3 grouping. And so since we have a carbon and hydrogen together, that's a non-polar bond. And actually the methyl group is your only non-polar functional group. So we do have other functional groups. You have the hydroxyl functional group, which is an oxygen and a hydrogen, which of course is polar. You have your carbonyl, which is a carbon double bonded to an oxygen, which is also polar. You have your carboxyl, which is a carbon double bonded to an oxygen, bonded to an oxygen and a hydrogen. So it's a carbonyl and a hydroxyl making the carboxyl, which is also polar and acidic. You have a amine group or amino group, which has to do with this an NH2, which is going to be also polar and basic. You have a phosphate, which is PO342-, which is going to be polar due to the charge. And it's going to be important when we talk about ATP, because that's the phosphate in adenosine triphosphate, ATP. So that'll be important there. The sulfohydryl is important because of the fact that, again, that's a polar, just similar to hydroxyl. But we're going to see that in our proteins. And then back to the methyl, CH3, it's not only nonpolar functional group, and you're going to see it when we are trying to bond it to DNA to condense it. We don't really need to worry about the condensing of it yet. At this moment, you just need to know that methyl is nonpolar. And other than that, we'll talk back about the methyl group when we get into transcription and gene regulation. So let's get rid of that question. And let's see. Which unit has a focus on enzymes? If you look at technically Unit 3 in the AP standards is where enzymes is. I go ahead and teach enzymes now because I find it most logical now with the fact that we're talking about proteins. Logically, we should talk about enzymes, which are type of proteins. Enzymes are catalytic proteins, so they're going to speed up a chemical reaction. The way they speed up that chemical reaction is that they're going to lower the activation energy. By lowering the activation energy, you allow for the reaction to occur more frequently, which of course increases the reaction rate. So big example would be if I had a cup of water and I had sugar in it, sooner or later that sucrose will break down into glucose and fructose. But it's going to take a while. But if I add the enzyme sucrase to it, then it can speed up that reaction, and then you would have the glucose and fructose much quicker. My students are going to do a lab this week in which they're going to use an enzyme called Catalase to break down hydrogen peroxide. And so hydrogen peroxide is H2O2. So they'll break it down into water and they'll break it down into oxygen. And you guys have seen this reaction. If you've ever gotten a cut and you put hydrogen peroxide on it, you know how it bubbles up and foams? That's because of the fact that your cells have catalase in them and you've damaged your cells, and so there's catalase out there. And so as you add that peroxide to your skin to try to clean your wound, the catalase is speeding up the reaction of the peroxide changing into, well, being broken down into water and oxygen. So the reason why it bubbles is because of that oxygen that's generated. So I guess because there's no other questions, I'm going to keep rambling about enzymes. So enzymes can be denatured. And the way you denature enzymes is that you change the pH. So each enzyme has like a optimal pH, and they can go about 1 on either end in order to stay kind of still functional. So good example for this would talk about your milk. So in your milk, you actually have bacteria. And so the bacteria is eating the sugar lactose that's in your milk. And they're releasing an enzyme called lactic acid, which we'll learn about lactic acid in Unit 3, so no worries there. And so as that lactic acid builds up within the milk, the pH continues to lower. And so sooner or later, that pH gets so low that it denatures the protein casein, casein, that's in your milk. And so when it denatures the casein, it turns it from kind of a soluble material into insoluble, which is why your milk gets a little chunky when it's old. And the reason why they call it souring is because of the fact that lactic acid is building up, which makes the milk taste sour, because acids taste sour. Another way that you can denature proteins is by excessive heat. I think some of you have had chemistry, and so if the temperature gets lower, then molecules just move slower, and that's not going to break a bond. But if you increase temperature, the molecules start moving faster, and they start vibrating. And because of this higher kinetic energy, it causes these bonds to break. So an example you have of that would be albumin that's in your egg. It's that kind of colorless jelly stuff that you find when you crack your egg open. It's in the egg whites. And so when you cook it, it turns from kind of the colorless thing into a white solid. And that's because you've denatured the protein albumin. So I think I've answered your question about enzyme, and I rambled for a little bit about enzymes too. So are enzymes not denatured by proteins? Enzymes are denatured by temperature. I'm saying that they do not get denatured when you decrease temperature. If you decrease the temperature, you're just slowing down reaction. You do not denature your protein. If you increase temperature and you continue to increase it, and it gets excessively high, then you will denature your protein. And we are supposed to talk about that there are some proteins that can reform. And the biggest example I can think about of an enzyme that's able to be denatured and then reform would be in hair. And so with my hair, if it was curly, right, that's due to a disulfide linkage. It has to do with that there's two sulfurs that bind to each other, which causes the coil in my hair. And so if I take a straightening iron and I straighten my hair, that heat is going to denature those proteins. But then when I go and I take a shower, I'm allowing those bonds to reform. And so I can actually remake my coils in my hair, or remake the curls. And a similar thing happens when you are making coils in your hair, is that you can go and you can add heat, which then makes the curl. And then when you wash your hair, the curl comes out because you, of course, broken those bonds. So I hope I've answered your question. And let's double check, see if there's any more questions in here. Okay, there's no more questions that you guys have put. So if there's no other questions, then I hope you guys have a good day. I hope this was helpful. And of course, I will see you guys again when I do my Unit 2 review. If you guys think that this is something that you want to keep doing all semester. If you don't want to keep doing it, then of course, I won't force you to come watch my lives. But I figured I would be available for you guys however I could be. So have a wonderful day if there's no questions. No questions. Bye, y'all.